Namaskar everyone. Myself Dr. Namrata, Assistant Professor from Department of Genetics and Plant Breeding, Agriculture Research Station, Mandor, Agriculture University, Jodhpur. First of all, I would like to thank organizers of Third Virtual International Conference on Advances in Food and Agriculture Science and Technology for providing me the opportunity to present my research work at this platform entitled Diversity and Principal Component Analysis in Fenugreek Genotype. Let's start with the fenugreek crop. Fenugreek is a seed spice used to enhance the flavor and nutritive value of the food. On the left corner of the slide, you can see there is the morphology of fenugreek plant displayed. The fenugreek seeds and leaves are utilized in preparation of Ayurvedic medicines from ancient times in India. In the right corner of the slide, you can clearly see there is the graphical representation of area, production and productivity of fenugreek in India from the year 2017 to 2021. The low productivity of fenugreek in India can be primarily attributed to the lack of suitable high-yielding varieties for different agroclimatic seasons, and development of varieties requires proper screening and evaluation of germplasm lines to assess their potential as suitable parents and providing the necessary variability for the success of breeding program. Keeping all these points in the mind, this experiment was planned using 71 fenugreek genotypes, including 4 reference checks, that is RMT143, RMT305, Ajmeri fenugreek 2 and Ajmeri fenugreek 5. These genotypes were received from different geographical regions and were evaluated in augmented block design with 5 blocks during Rabi 2022-23 at ARS Mandor, Agriculture University, Jodhpur. Each genotype was accommodated in single row plot of 3-meter length with a spacing of 50 cm between row and 10 cm between plant. A total of 10 phenotypic traits were observed, which is enlisted in the slide, you can clearly see. The multivariate analysis of D-square was done for all these 10 characters by using Mahalobin statistics, and different clusters were formed by using Ward's method as described by Rao, and principal component analysis was performed using RStudio. Now moving to the results. The clustering pattern of fenugreek genotypes revealed that the study material in this experiment can be grouped into 3 diverse clusters. The largest one is the cluster 3 containing 31 fenugreek genotype, followed by cluster 1 that contains 26 genotypes, and smallest one is cluster 2 that contains 14 genotypes. The maximum genetic diversity present between cluster 1 and cluster 3. So the crossing between the members of these two clusters helps in exploiting high heterosis. Here the cluster 3 showed maximum variability within the cluster because it included exotic collections from NBPGR Jodhpur and germplasm from SKNU Jogner also. Now coming to the dendrogram. The clustering pattern revealed that genotypes from the same origin may be present in the same cluster, like the AM280 and AM281 present in the cluster 1, and both are originated from Ajmer region, while it may be present in the different cluster, like AM280 is present in cluster 1 and AM108 present in the different cluster. So geographical origin can't explain about the diversity present or the vice versa. Now coming to the principal component analysis. It was calculated for the database comprising 71 fenugreek genotypes, including these 10 phenotypic traits. The spree plot presented here clearly show that the components having an eigen value more than 1 were selected, and 5 primary components contributing most significantly to the overall variation were identified. These 5 main these five main components contributed These five main components contributed a cumulative variance of 76.51%, and out of which principal component 1 explained 28.1% variation, followed by principal component 2 that explained 15.11% of the variation, which is clearly demonstrated in the table. Now coming to the biplot. So the biplot visualization allows for diverse interpretations. The traits with the longest vector in the direction of principal component were 1 were the most influential, that is plant height, then seed yield and biological yield, while principal component 2 was favorably impacted by pods per plant, branches per plant, days to maturity and days to 50% flowering. So in the concluding remarks, we can say the principal component 1 can be interpreted as a dimension of overall crop vigor and productivity, and the breeding programs that aim to maximize plant height, seed yield and biological yield should focus on moving genotypes towards the positive end of the principal component 1. While principal component 2 can be interpreted as a dimension, distinguish between early versus late maturity traits and yield structure. So the breeding programs with the objective of traits, early flowering or a higher number of pods per plant can use the insight from the principal component 2 to select the appropriate genotype for being the parents in the crossing program. With these remarks, my presentation is over. Thank you very much for your patience. Thank you.